Great. Thank you so much. Okay, so my talk is going to be about environmental microbiomes, and more generally, my lab is interested in the concept of human-environment microbiome interactions, particularly in the context of antimicrobial resistance transmission networks. So we are interested in developing new technologies to study this. As Sanjay mentioned, some of these challenges that the current technologies and the current databases are not adequate. And so we try to use longer read genomic sequencing technologies to get at higher resolution analysis. And also in being able to model the microbiome. And Sanjay also talked about this concept that we need to understand how these various microorganisms are interacting in order to understand their function. We are interested in the skin microbiome as an interface between the environment and human microbiomes. We are also interested in the impact of antibiotics on the gut microbiome. Sanjay talked about antibiotics in the lung microbiome. We worked with Barnaby in the past to look at how the antibiotics, and Yunn as well, how antibiotics disrupts the gut microbiome and how the microbiome recovers. And in recent work, we're looking at CRE colonization of the gut microbiome. And finally, we're interested in various environments beyond the hospital. So we're looking at food courts. We're looking at surveying Singapore in general and getting a microbiome map of Singapore. Okay, so all of this work is really motivated by a challenge that we're all well aware of, which is the global antibiotic resistance epidemic, and the fact that it can have a huge impact on human health and our economies if it's left uncontrolled. So this problem is already a severe problem in various parts of Asia with very high incidence of resistance to our last resort antibiotics. And a lot of focus in terms of reducing AMR rates is in terms of antimicrobial stewardship, in reducing patient-to-patient transmission in hospitals, reducing the risk of outbreaks. But relatively less is done, at least at the genomic level, in terms of looking at environmental reservoirs and how they can serve as a node for transmission. So there's been some initial work looking at what we call the hospital microbiome as a reservoir which can be seeded by patients and then seed patients back in time with many of these multi-drug resistant bacteria. Now, the initial studies, and this is some pioneering work from Jack Gilbert's lab, looked at really the question of what happens when a newly built hospital gets colonized by bacteria over time as patients come in. And they were looking at it from an ecology perspective, like what are the community dynamics over time. And they used a very coarse profiling approach, a 16S ribosomal RNA profiling approach, which lets you only get information at high taxonomic level. So these are bacteria which are separated by millions of years of evolution, not the specific pathogens of interest for us. In more recent work, they've looked at neonatal ICU units. Obviously, these are areas of importance because of at-risk patients in there. And they use short-read metagenomics. So this allows you some strain-level analysis, but it still does not give you the whole genome information that we need to understand transmission pathways. So we decided to embark on a project together with collaborators here in Singapore and clinicians who were really interested in understanding what are really the reservoirs, can we do them at the whole genome level, and really over the whole hospital, not just in specialized units. So we selected a set of beds as well as kinds of wards, and also looked at sites which are high-touch sites where we already know there's concern about transmission through these sites. So these include things like the bed rail of a hospital, of a bed, or the door handle, or sinks in the wash basin, or the aerator caps. So different sites that are of concern for environmental transmission of bacteria. We used a traditional approach, the Illumina shotgun metagenomics approach, as a first step, and we used this to characterize 355 microbiomes from 179 sites, using a 2-time point approach. So we wanted to understand the resilience and the robustness of the microbiomes. This is 1 week apart. But in addition, we use a time point which is a year and a half apart. So this is to establish long-term persistence in the microbiome. And we used about 30 million reads on average. So this is relatively deep sequencing for shotgun metagenomics. So one of the first things we looked at is really the notion of ecological niches in the microbiome. And this is important because, as Sanjay mentioned, the communities of bacteria that are present in a particular site might define which pathogens find it easier to colonize in a particular site and how persistent they are, if they form biofilms, and so on and so forth. And so when we did this sort of analysis, here what I'm showing you is what's called a PICO-A plot, which is taking the taxonomic profiles for each site and compressing it down to a dot on a two-dimensional space. And what you can see from this figure is that there seem to be two different regions of high density of dots. And the two different regions correspond roughly to two community types in the microbiome space. The assumption here is that the higher the density of dots, the more likely that's an equilibrium configuration for the microbiome, for the profile of bacteria that are seen at a certain site. So we call these community type A and B, and we're using these as entities for further study. And what we find is that community type A is primarily human contact sites. So these are the bed rail or the bedside table or the cardiac table below the patient's bed, and so on. So these are regions where people actually touch the surfaces quite often. The other sites, the community type B sites, tend to be what are typically areas of concern for infection control. So these are the aerators and the sink traps, the more kind of wet sites which have flowing water, but we may not necessarily touch directly. It might be through water that bacteria are transmitted. And the other thing we noted was that even though they have these two distinct sites, when we compare them to more than 1,000 samples that we have collected all across Singapore, from parks, from playgrounds, from workplaces, the microbiomes that we see in these other sites shown here in blue are quite distinct from what we see in the hospital environment on hospital sites. So this suggests there's something happening in the hospital, and there are many hypotheses of what could be happening, including the cleaning measures, the fact that there are patients with various infections and various disease conditions in there, which seeds and creates a different environment in hospitals than you would find in say, in this convention center or in the offices that we work in. So in terms of these distinct community types, they also exhibit different profiles of bacteria, not surprisingly, because we'd identified them from the profiles. And the two distinct profiles are really defined by, the community type B is defined by biofilm-forming bacteria, while community type A is defined by skin bacteria, which are presumably being seeded by contact with humans. So this provides us a defining foundational knowledge of how to think about hospital microbiomes. The sites which have more human contact and are shaped by human contact, they tend to be more dynamic. So this plot shows you the same two-dimensional plot, but now two dots are connected by a line if they're from exactly the same site but a week apart in time. And you can see that the various sites which are not in the blue box, they tend to have a lot more changes over time, while the sites which are in the blue box, which are coming from community type B, the biofilm forming sites, they are a lot more stable communities. Despite all the cleaning measures and all the efforts we do to reduce the burden of bacteria in the hospital environment, these persist stably. So one of the goals for this project was to really understand the utility of such metagenomic profiling and surveillance for infection control. And of course, one of the interests is to understand what is the distribution of antibiotic resistance genes, in particular the beta-lactamase genes. And these are readily detectable from just shotgun sequencing data. So that's already a win. And in addition, we show that you can use the data to compute various scores which could be of use. For example, things like how much human influence, how much human DNA we can find on these sites, suggesting that they have greater human contact. How specific is the microbiome of a particular site in relation to the microbiome nearby, so how much transmission might be happening. And also how much turnover from time point to time point, how much turnover is happening in a certain site. So all of this perhaps can inform infection control, how frequently we should survey and how frequently we should clean a particular site. But when you start thinking about the specific pathogens of interest in infection control, the most common pathogens, the escape pathogens, these are really low abundances in the microbiome of any particular site. So just Illumina shotgun sequencing of, say, a swab from a site is not going to give you detailed genetic characterization of that species. So to address this, we used a culturing approach, but it's not fully culturing. So it's what we call quasi-metagenomics, where we do a bit of culturing and perhaps use antibiotics to select the multi-drug resistant bacteria. And then we use a plate sweep approach to collect all the DNA that survives this very short duration culturing approach, and then sequence all of that DNA to understand the genetic component in there. The advantage of this approach is that instead of just getting a few reads, which might hint at the presence of a certain bacteria, we can actually do what's called assembly, which is piecing together the reads into constructing a whole genome. So this is a computational reconstruction process, but it's really aided by the reduced diversity that you get when you do a little bit of culturing and do some antibiotic selection. So we get more than 2,000 high-quality genomes from a few hundred sites. So this is a very large collection of genomes, and as people who work with infectious diseases and doing epidemiology, genomic epidemiology, you know that whole genomes are really the gold standard for studying transmission networks. So in addition, we get plasmid information, again, very important to understand what is the reservoir of plasmids which can serve as the backbone for transmission of various resistance genes. In a symposium tomorrow, I can go into greater details about how we do this assembly and how we do this sort of analysis, but I will just assume that we have these genomes through a computational reconstruction process. And one of the things we noticed was that we got cultures from a lot of plates which have antibiotics on them. So we suspected we would get, say, 10% of the plates would have cultures, but in fact we found that more than 60% of the plates have cultures, more than 50% of the sites have bacteria that are resistant to all 5 antibiotics. So we get cultures on all 5 plates. And really we get a diversity of morphologies and phenotypes, including hypermucoviscous kind of phenotype. And so when we look at this data, this very rich data, we realize that these are genomes which we could not have acquired without doing this intermediate culturing step. Even though the media was not really selective media, it really helped enrich a lot of these pathogens. And the genomes we acquired, as shown on this graph, the N50 is the median size of the contigs we assemble. And as you know, a microbial genome is about 4 megabases long. And as you can see, the median N50 is typically several megabases. So typically, we get a genome as one piece rather than many, many small pieces. So this is even better than what you would typically get from taking an isolate and doing Illumina sequencing on that isolate. So now that we have genomes, we can do the classical investigative work, epidemiological work that we can do with whole genomes. So we took the Staph aureus genomes that we have from various environmental sites in the hospital and related them through a phylogenetic tree. And what I'm showing you here is the phylogenetic tree condensed at 99.9% similarity. There are many, many more strains. I'm just collapsing them at that threshold at strain level. And then also overlaying the antibiotic resistance information. Where was these strains, which plates were these strains obtained in? And you can see that there are some strains which seem to be resistant to all 5 classes of antibiotics that we had here. And in fact, when we look at these same strains and their distribution across the hospital, so now we have actually a map across the whole hospital, we can find that these strains are the strain S1, which is marked here, being resistant to all 5 antibiotics. I guess I didn't realize that you can't see my pointer. So the strain S1, which is resistant to all 5 antibiotics, it's also seen distributed all across the hospital environment. And it's also persistent. So the 2 different colors that you see here, orange and blue, show you the, thank you. The 2 different colors, which don't seem to appear very well in here. I'm sorry about that. The orange is the first time point and blue is the second time point. You can see that the same strain here, S3, is persistent in both time points and it's seen in many different sites in the hospital environment. So this was kind of intriguing to us, and this is analysis with Staph aureus, but we have genomes of many other species. So we can repeat this analysis across a range of genomes where we have sufficient genomes, and you can see that this pattern is seen again and again. Whenever we see something that's multi-drug resistant, it also seems to have a lot of different connections. It seems to be present in many sites in the environment. And we can assign a statistical significance to it. This is actually a real pattern, which is not by chance. So that's quite interesting. There seems to be some reservoirs, and these reservoirs seem to be enriched for multi-drug resistance. So why is that happening? And we sought to look at the potential sources for these resistance cassettes. So now, since we have whole genomes and not just fragmented genes, we can start to look at cassettes of antibiotic resistance which are there in these genomes. And so we use that as a unit of analysis. One of the things you observed was if you looked at all the antibiotic resistance cassettes or pairs of antibiotic resistant genes, more than 24% of these gene combinations are novel. They've never been seen before in our databases. Right? And then even more, sorry, for some reason my click is not working. Sorry about the technical issues. Okay. So for, oops, go back a bit. Okay. Right. So 24% are novel. And if you look at just the chromosomal ones are very stable. So the resistance cassettes, the set of genes which are there in the chromosome tend to go together all the time. They don't move around between species or between cassettes. But then when you look further into cassettes which are there in plasmids, these seem to move around quite a bit. So here the circles indicate plasmids, and the dots indicate the resistance genes. And a line between a dot and a circle indicates the relationship between the resistant gene and the plasmid. And as you can see, many resistant genes are present in many plasmid backbones. So this suggests that even though the presence of pathogens in the environment is quite robust, and they are quite often multi-drug resistant, the resistance genes might actually be being shuffled around in the hospital environment. They might be moving around through plasmids in various plasmid backbones. So more than 90% of these plasmid clusters that we had, they have never been seen before. And in fact, we could find some very concerning plasmids. One plasmid backbone was seen in MRSA and has biocide resistance genes, again not been seen before, and multiple antibiotic resistance genes. So this suggests a mechanism by which we might be selecting for this resistance through the cleaning measures that we might be employing in hospitals. Okay, so this is all just in the environment, right? We didn't have patient isolate. So what happens when you combine this with patient genomes, patient isolate genomes that we have acquired through surveillance efforts? So this is data from an MRSA surveillance effort, which was done nearly a decade ago in Singapore. And we try to relate these genomes to the environmental genomes. And the colors here indicate whether this was seen in the hospital or in a patient. And you can see that some of these genomes were indeed, these strain genomes, were indeed seen in hospitals and patients, suggesting that there's some sharing, and in which way that sharing is happening is not clear. But remember, the patient genomes were acquired nearly a decade ago, and the hospital environment genome was much later, suggesting that there is a reservoir, and that reservoir is the basis for these infections. So because we have multiple species, we can do this analysis again and again and again. So we again and again find that when we find a sharing between the hospital environment and patient genomes, these genomes tend to be multi-drug resistant, or seem to have the multi-drug resistance phenotype based on both our culturing as well as the resistance genes that we can see in these genomes. So I want to summarize by saying that we think that hybrid shotgun metagenomics, the idea that we can use Illumina sequencing for profiling the community, but use culturing and nanopore sequencing to get whole genomes. This is a powerful way to monitor hospital environments and figure out where are the areas of concern. We see distinct ecological niches in the hospital environment, suggesting that we might have to tailor our interventions according to these niches. They have different stabilities and different resistance genes that can be observed in there. And there are some observations which are contrary to what we have assumed before. For example, Klebsiella pneumoniae has been typically assumed to be something that's present in aerators and sink traps and so on. But in our study, we find that it's more abundant in community type A sites, which are the drier, more high contact sites, suggesting that culturing-based approaches might be missing things that sequencing-based approaches might pick out. They might be dead cells, but they are likely to have been alive at some point of time. And in that particular environment. So in addition, we think there's a substantial understudied diversity of resistance genes and resistance cassettes. And having a clear picture of what that diversity is important to understand what could come next. What's the next multidrug resistance plasmid that could be disseminating through the hospital environment silently till we start seeing infections that affect our patients. And the last point, which I think there are many possible mechanisms by which this could happen, but it's still an interesting observation. The first time this has been established is that there seems to be an enrichment of multidrug resistance in hospital environments, and this is also associated with persistence in multiple locations and over time. So this work is freely available through bioRxiv, which is kind of a public repository, but it was also published in Nature Medicine last year. And the scripts and all the analysis that we do is freely available. We're happy to share our data as well as our scripts. So I really want to acknowledge a lot of the work that was done by members of my lab, Kern Ray, who really led the project, Chen Hao and Amanda, who were a great team working together, along with Denny and Samantha here on the right, and our clinical collaborators, Kalis, Untek, and Brenda, who really initiated this project. So all of this work was done in participation with the Metasub Consortium, the Global Metasub Consortium, which is interested in environmental microbiomes and their influence on human health. And for us, really, this was a first step in a vision for the future where we are doing infection control, not blind, but using genomic technology to see what we are trying to control. So real-time tracking and targeted eradication of antibiotic-resistant pathogens. So I'll leave you with this video, if it actually plays, which is our view of how things could be in the future. We took our data and we thought it'd be fun to use this sort of video game creation software to visualize the data. And we sort of imagined the future, people be walking around with augmented reality glasses, and they can just walk around and point to a site, and the data that we have about the specific microbiome profile, the pathogens, the persistence of bacteria in that site can be put up. And then infection control specialists can perhaps figure out which sites seem to have connections, as in they seem to have the same strains, and then maybe target the infection control to those sites. So this is just a depiction of data from our study where we showed that Klebsiella pneumoniae in one of the sites in the bed rail of a patient was exactly the same strain, multidrug resistant, which is also seen in the sink trap of that same room. So this sort of information could be very useful in the future. We just need ways in which we can convey that can be useful to the clinician. So with that, thank you for your attention. I apologize for all the technical hurdles, and I hope this was interesting to you. Thank you.
Great. Thank you very much, Niranjan, for the talk. We have a couple of questions from the floor, which I hope we have some time for. So let's first the question here from Ichi Xiao. So it said, very interesting, comprehensive work. Are you able to differentiate between live and dead cells, cell-free versus cell-associated plasmids in your samples?
Yeah, so the short answer is no, because we're doing everything using DNA sequencing. But the quasi-metagenomic work that I described has a culturing step and has an antibiotic selection step. So anything that we sequenced there was at least alive in our culture. So, and we have various controls to show that if you just, these are not laboratory contaminants. So if you just take a blank swab and put it on our plates, we grow nothing. So we strongly believe at least the quasi-metagenomic data is talking about live cells acquired from the hospital environment. The second part of the question about plasmids, that's also an interesting question. Again, for the quasi-metagenomics, we can be confident that these were in some cells at some point of time. For the Illumina shotgun metagenomics, we have no idea if they were ever part of a cell or not.
Okay, yeah, thank you. We have a question here from Crystal Wong, which I think is an interesting question, because you've looked at the environmental microbiome and the patient microbiome, and to sort of then extend that to the third group, which would be healthcare workers or other people working within the environment or visitors. Have you looked at this group? And then also, sorry, then how that might affect the microbiome, the environmental microbiome within the home of people with healthcare workers?
That's a fascinating question. So I should clarify that the patient data we acquired was acquired from studies that were already published out there in the public domain. It was not done along with this environmental transmission study. And there are obviously ethical considerations to take into account while doing this sort of studies with patients. As a next step, we are indeed trying to do surveys where we have patient samples and patient microbiomes at the same time as we get the environmental microbiomes. So I think that's the next step. And the step after that, of course, with healthcare workers is fascinating and interesting. But the ethical considerations are the ethical barriers perhaps even higher to convince people to provide samples. So I think once we establish that the environment and patients are really interacting heavily, we could go the next step and talk about healthcare workers and their influence on the microbiome.
Okay, cool, great. Thank you very much.